Good afternoon, everyone. Hello, sleepy, energized, reignited? No, still need some more time to settle up. Can we start? All good? Was the snacks good? Did you have? Okay. Before peeping into the topic, uh, it echo. So directly peeping into the topic, let's just glance about two sessions what happened before. Mr. Sunil Kumar came. It took us with a network pharmacology from a drug to a protein, how to select a protein, how to exactly get into a protein, and followed by session, Mr. Vasangoda told us what is protein, what is ideal protein, how to get into a protein, like how to download a protein. What if we don't get 3D structure of a protein? So what to do? Any idea? We'll all do research work and all. We'll end up to a target. We'll get into a protein target, and we'll see in the PDB there is no protein 3D structure. What to do? Any idea? No. So there is a solution called as homology modeling, where we'll build or predict the protein structure. So these are the contents, introduction, what base about protein structure prediction, what are the steps involved in protein prediction, application, pros, cons, and we'll see the demo. So what is homology modeling? So it is a computational technique in silico method where we will build our desired 3D protein structure using a template. So I'll be walking around, I'll see a good home. I want to construct the same similar home. I'll give a what I need to do. Need to go to a contractor and ask for the like things what all I need to do. He'll give a blueprint. Then the similar structure, like similar, I can build the home. Similarly, after research, we'll get a what do you call it? Homologous template we'll find with our query structure, where we can build three-dimensional 3D protein structure without usage of X-ray crystallography and NMR crystallography. So we need to find significant sequence similarity to have structural similarity. Exactly, I need a 3D structure of the protein. I keep on searching. At the end, I'll end up with a similar sequence of protein, which will be similar to my sequence. So before start building into a protein, what exactly is protein? We got two sections about protein, sessions about protein. So what exactly protein is? Any idea or anyone? Two sessions were bad? No. Sir, daily I go to gym, I take protein. That is the protein. Anyone? No. So protein is a linear sequence of amino acid. Just sequence of amino acid make up a polypeptide chain that is the base of a protein, followed by a local three-dimensional structure such as beta helix and alpha sheets, where this linear sequence of amino acid stabilizes by hydrogen bond, where it leads to a 3D structure called a secondary structure. It is not like that two primary structure gets into secondary structure, two secondary structure makes up tertiary structure, two tertiary structure make up into quaternary structure. It is not like that. This beta helix and alpha sheets are formed by this linear sequence of amino acids, followed by tertiary structure. Overall, three-dimensional structure of these amino acids, which form a linear polypeptide chain, these have different part where interactions happen. So, including hydrogen bond, ionic interaction, and disulfide bridges are there. So, overall, this three-dimensional structure, quaternary structure is the final structure where it is crucial for protein functioning and many properties of the function. So, quaternary structure, exact structure or the like what called as up to the mark, what do you say? If we have the up to the mark quaternary structure, we will have a better protein for our studies. So before building a protein, if I have a similar sequence protein structure or a sequence of amino acid, I can get into a or search for a template in protein database. What if I don't have? If I have, I can go with homology modeling. If I don't have protein sequence, or if I don't get protein sequence, I can go with ab initio, de novo. From the scratch, I need to start building up the sequence of amino acid to make up a 3D structure. In threading, another protein prediction method is there, threading, where folds will get folds of a folds of proteins are there, where we'll compare that with our target protein. With comparing that target protein, we'll build up our own 3D structure. So these are the some steps. We'll getting into homology modeling, template selection, backbone generation, loop modeling, followed by sidechain modeling, model refinement, and model validation. So template selection involves like what I told before. Before getting into like I need to construct a home, I need to search for a blueprint. That is the template. I have a query. I need to keep searching for template that will get in UniProt or protein data bank anything. So followed by after getting that UniProt ID, we'll blast the sequence. Blasting nothing but will not break the protein structure. We'll build up the protein structure. That linear sequence of amino acid is built in a line. Choosing a template with high resolution is important here. Not more than 5 Armstrong as sir told. Below or nearby to 2.5 Armstrong is ideal for the work. So we can see here. First line is my query template. Second line is query. Second is template, where some of the residues are missing here. I got a query template. I keep on search. I finally found a template in protein data bank. Okay, this is a simple example. So some of the criteria are there here. Sequence identity, where at minimum 30% or more than 30% sequence similarity should be there, which is good for building up a protein. Below 30%, there's a chances of perfectly going wrong. We'll never end up building a protein. Structural resolution, more equal or more than lesser than or equal to 2% Armstrong is better for building up a protein structure. Sequence coverage, fully or nearly full alignment. Here, like 80-85% sequence are completed. Only some 3 parts is missing. So it's okay. Like again, we can build it in backbone generation or in loop modeling. We can build up this sequence up. If more number of residues are missing, that is like final structure of protein will be invalid. Secondary structure, we should look up for alpha helix and beta sheets. So these are the more important structures for building up a protein. So the database validation can be done in protein database. Second step is backbone generation. Backbone generation is like constructing up the main of a protein. So backbone is sequence generation, where target protein based on alignment with looking up with the target template will start building up the same thing. Protein is nothing but a combination of amide group, carboxylic group with a central carbon. These 3 nitrogen, alpha carbon and carbon is crucial for developing a backbone of the protein. So 2 major things which should be kept in mind: structural stability and poor electron density, where it should not end up falling in a like wrong manner. We'll be having what do you call poor sequence, poor buildup of sequence or lack of sequence, which we found in this template thing. Followed by challenges what we see in the backbone generation. Gaps in alignment, more number of gaps where we'll end up in a bad protein building. Conserved residue misfits standing in a wrong place, where a particular amino acid should be standing in a particular sequence. It will not be at a particular place, where we will not end up building up a proper protein. Backbone steric classes, which may lead to steric hindrances, where energy will be high. Again, the loops and helixes may not form properly. Low quality template regions, where proper sequence when we are not maintaining, again you'll end up with a low quality protein or building up a 3D low quality model. So some of the tools are there. Modeler, Swiss Sidestein and Rosetta, where we can build up a backbone for a protein. Followed by loop modeling, which deals with predicting the conformation of a loop regions in the protein, which are often variable and do not align with the template. So loop are a segment in a protein where 2 secondary structures are aligned together. where we need to build up or make a loop. Accurate loop modeling is essential as loops can be crucial for protein functioning and uh, interactions. So secondary structure like alpha helix and beta sheets are there. Proper torsion angles may lead to like extreme electron densities, and again steric hindrances will be there. As I told, like challenges are there, flexible and long loops, where we can be used molecular dynamic simulations to handle the conformations of the flexible and long loops, where flexible loops in the sense protein amino acid sequence will not be aligned particularly or properly, where it interacts, and again flexible loops will be there. So basic of the protein is amino acid, where amino acids are not properly synthesized and not properly sequenced. So basic amino acid is responsible for building up alpha helix and beta sheets. It will end up in a bad building of a protein chain. Incorrect anchor placement. Anchor is nothing but protein, membrane protein, which helps us connecting to the cells. So some of the tools are there: loop modeling, prime for loop modeling, prime by Schrodinger, Chimera and Rosetta are the things where we can build up a loop for a protein. So side chain is there, involves like where to a backbone side chain is present. Alpha carbon and nitrogen atoms are there, where connected with the for this amide backbone, side chain is built up. So this technique minimizes the reduces the energy minimization, where itself help us to build up a proper protein structure. So again coming to model refinement, where it is a sum of backbone generation, loop modeling, and side chain modeling, where we'll reduce the errors, improvise the initial structure, and optimize the structure resolutions. Some of the tools are there for model refinement: Gromac, Amber, and Squirrel Co. Model validation. After doing all these things, we need to validate the model to check the quality and reliability of the model. So it is done for its geometry check and energy profiles. So this can be validated using Ramachandran plot. So some of the key metrics are there. Ramachandran plot, percentage residues in the favored region. As we saw in the previous session, there was two colors, red and yellow. The more number of amino acids present in the red region, like it favors the protein structure. More than 90% should be there. RMSD, root mean square deviation, it should be lesser than or equal to 2 Armstrong. And we should verify for the 3D structure. 80% compatibility should be here. So some of the applications are there: drug discovery, where we can identify a potential drug targets for the existing drugs; protein function prediction; uncharacterized proteins by we can compare them. A known protein structure with unknown protein structure, we can similar sequence if we have, we can predict the function of the unknown protein structure. Protein engineering, evolutionary studies, and antibody designs also can be done using this. Some of the pros are cost effective. It is significantly cheaper and faster than experimental method like X-ray crystallography and NMR. For example, there is a technique called cryo-EM, where for a single building up of a single protein structure, we need up to 8,000 movie stacks, followed by like 10 to 15 TB of memory and all. It takes long process and all. If we have a template and a query protein sequence, we can just by comparing them, we can build up here. Cost effective and less time consuming. Insightful predictions provides valuable structural insight for protein without known structure, aiding its functional annotation and for the drug design. Versatility, it can be applied to a wide range of biological problems here. Not only restricted to Homo sapiens, it can be used for in animals preclinical studies also. Accuracy limitations, where it is not exactly as we find in the X-ray crystallography or NMR, like it is lesser accuracy when compared to those things. Unreliable for novel folds. We will not be able to find the new or the novel folds of the protein 3D structures here, sensitive to alignment errors. Properly not aligned things get mugged up here. So we'll with a known pro uniprot ID, we'll just build up a protein 3D structure here. With a tool called Swiss model, we'll build a protein structure. From a free protein 3D buildings software called as Swiss model, we'll build a 3D protein structure. From the protein database called as Uniprot, I have a ID. On clicking start model, we can paste the target sequence here, or if you have a Uniprot ID, we can paste here, or if you have a notepad or a Word document, we can upload here, but we cannot upload screenshots here. If you are a member of a deep view project, directly from deep view project, we can upload the things here. User template, if you are not properly sequenced or built 3D protein structure is there, we can upload here. I have a Uniprot ID. I'm clicking validate, so it aligns here. Automatically the name took here. We can add email ID is optional. Without logging into this, we can get into here. After that, we need to search for templates. So this procedure may take up to 15 minutes for some templates. Till then, other than this, we can use AlphaFold mod, Swiss model, alternative for Swiss model, or Rosetta Fold can be used. So coding like Python can be integrated into AlphaFold for building up a 3D structure of a protein. Also. Hmm? Off, off. Alright. Minutes. So menu appears here after a template is loaded. There is options called as template, quaternary structure, sequence similarity, alignment, and more. This template is called in a green color highlighted 50 they shown. The mentioned ID from Uniprot, it has searched for in the protein data bank, it has searched for 50 protein IDs which are similar to my query structure. It has automatically selected one. A blue line appears here where it's showing coverage 1. The values varies between 0 to 1. the nearer to 1, the more similarity sequence is there. So it is exactly one, it's similar, like most, it is exactly similar to my structure. And another thing is GMQU, QSQE, and identity. Global mean, I'll tell about this. GMQU, QSQE, and identity and method. These four things should be pointed out here or noted down here. After selecting the required gene like target template, we can click on the build models where it will start building the 3D structure of a model similar to our query sequence. So to validate the 3D structure, structure assessment is there, where automatically it generates a Ramachandran plot where we can validate the protein structure. Before that, so there are four quadrants here. This region tells about the beta sheets, and here alpha helix, and bottom right tells about the alpha helix. So in this standard plot, where we can see the red region and yellow region, the more number of amino acids present in the red region is favorable for us. Okay, okay. In Swiss model, they have ಇದು ಹೋಗ್ತಿಲ್ಲ ಸರ್. ಇದಲ್ಲ ಈ ಸ್ಕ್ರೀನ್ ಬಂದಿಲ್ಲ. ಎಕ್ಸ್ಟೆನ್ಷನ್ ಕೊಡಕ್ಕೆ ಹೋಗ್ಬೇಡ್ರಿ ಅಂತ ಬರ್ಕೋತಾ ಇದೀನಿ. ಎಕ್ಸ್ಟೆನ್ಷನ್ ಕೊಟ್ಟು ಸಾಯ್ತಾ ಇದ್ದೀರಾ. ಎಕ್ಸ್ಟೆನ್ಷನ್ ಕೊಟ್ಟಿಲ್ಲ. ಎಕ್ಸ್ಟೆನ್ಷನ್ ಕೊಟ್ಟಿದ್ರ ನನ್ನ ಮಗನ ನನ್ಗೆ ಗೊತ್ತಿಲ್ಲ. ಶೇರ್ ಆಗ್ತದೆ ಡೂಪ್ಲಿಕೇಟ್ ಕೊಡು. ಬಂದು the dark green color region where more number of amino acid present, residues amino acid are present. Dark green region, more number of amino acid dots can be seen. It tells about the favored region. So 96.36% favored regions are there. And in the below section, some of the residues are there. So it tells about the alpha helix. Above region tells about the beta sheets. Which type of structure is present in our protein? Which type of primary structure is present in our protein 3D structure? It tells about this. The more number of amino acid present in the dark green region tells about this favored region. We can download this protein structure. There is an option called download file. We can download this 3D protein structure in the PDB format. And also we can look into this GMQE, which is global model quality estimate. It tells about the quality of the protein. And there is a which tells the quality of the protein. There is another QSQE, which tells the quaternary structure quality estimate, which tells about the interchain action, like alignment, the interchain action in the protein, 3D structure protein. How is that? It well explains in the QSQE. Q mean score is nothing but overall score for this quality estimate. So this GMQ and Q mean score varies between 0 to 1. The value nearer to 1, the more structural similarity with the template. Our query structure will be similar to our template. So we can download this in PDB format, and you can use for this like docking or dynamics. If you want to change the structure or a shape, we can use this element surface or any like cartoon thing to which is favorable for us, we can use this. So this is simple step where we can build 3D structure of a protein with our query sequence using template of a known protein. Thank you.